presentation is meant to inform about the purity and DNA content of AAV capsids and how these critical quality attributes can be assessed by analytical ultracentrifugation and orthogonal biophysical methods. Analytical ultracentrifugation is a very established biophysical method that has been used to characterize biochemical entities regarding their molecular weight, size, and sedimentation properties. When developing and manufacturing AAV-based vectors for gene therapy, it is important to have analytical methods at hand to assess the product quality. The aim of our study was therefore to compare a set of analytical methods to work with different AAV serotypes and align them regarding the information they provide. Different methods were selected for this purpose, including anion exchange chromatography, UV spectroscopy, SVAUC, electron microscopy, and mass photometry. And the focus was on the determination of empty and filled ratios and on the additional information provided by each method. Serotype-specific differences were evaluated as they were observed during the investigations. The different serotypes were characterized regarding concentration and A260 to 80 ratio by UV spectroscopy, structural integrity by electron microscopy, mass photometry was used to reveal molecular weight and insert sizes, charge variants were revealed by anion exchange chromatography, and SVAUC was applied as gold standard method to assess empty, partial, and filled species and high molecular weight species. Looking at the results for the different serotypes, it's evident that AUC does not distinguish strongly between the individual serotypes. We used AAV 2, 5, 6, 8, and 9, and the C over S distributions were highly similar for the empty capsids at around 65S, the filled capsids at around 95S, and additional information was provided regarding partial species between the two peaks of the empty and filled species. Anion exchange chromatography also separated the empty and filled very nicely for AAV 2, 6, 8, and 5, and not as well for AAV 9. It's important for anion exchange chromatography and AUC to keep in mind that the contribution of empty and filled species is dependent on the extinction coefficients, which are different for the two species, and therefore a renormalization has to be performed to come to capsid numbered based ratios. These capsid numbered based ratios can be determined by electron microscopy based on the individual staining patterns of empty and filled species, independently of AAV 2, 5, 6, 8, or 9. Also, these capsid-based ratios can be determined by mass photometry based on the different contrast the AAVs provide for empty and filled species. And in cases where both of them are present in the same sample, two peaks are observed at the molecular weights of empty and filled capsids. In the end, each of these methods provides empty and filled capsid information, in addition to the properties that are revealed based on the individual methods characteristics like molecular weight, insert size, capsid concentration, or impurity presence. Looking at the correlation of the empty and filled results between the different methods, it is clear that AUC and chromatography correlate very well. Also, AUC and mass photometry correlate very well. And also the correlation with the other methods in general is very good. Each of the methods provides thus information on the empty and filled ratio while providing the information that is specific for each method that we applied. Reveal that SVAUC can be applied orthogonal to other methods in a platform approach with consistent data obtained for the empty and filled ratios. The correlation is especially good with mass photometry and anion exchange chromatography, and this was also revealed for mass photometry if further species are present in the species. SVAUC provides additional information on background material and the concentration of the AAV samples, and the application can be done also as a GMP compliant method to investigate these critical quality attributes. With this, I want to thank the team that worked on this study and you for your attention.